Thank you. Good afternoon, everybody. Our research is in, let's see. Our research aims to understand how biological shape varies across individuals and is affected by different diseases, how biological shape relates to biological function. In the context of the brain, this means how the brain activity relates to brain anatomy, for example. So we have a wide variety of projects in this area. Today, I'll show you one example of the type of analysis that we might perform. And it has to do with modeling, anatomical variability, but again, for those of you who are interested, we're also working on connecting function with that. So specifically, the question of anatomical variability has been a big one when one is trying to build description of how a particular disease affects the brain. And traditional population studies all run using similar kind of prescription. You collect data, imaging data, so for example, MRI of the brain, from a normal control population and the population afflicted with a particular disease, for example, Alzheimer's or schizophrenia. And then you ask a question, what are the localized differences in anatomy that are present between these two populations? One of the challenges in that have come up over and over and over again is that the natural variability in each population is so large that the small subtle changes induced by the disease might be not high enough to be detected using this method. Oftentimes, the conclusion of the study is that, well, actually, it's not entirely conclusive because natural variability is so large that the differences are too small relative to sort of what we might call noise in the data, but it's a natural, very structured noise in the data. So our approach is to turn the problem upside down. And specifically, we will use images to define the populations. Given a large data set of images, we will find partitions in it of reasonably homogeneous subpopulations where the anatomy is quite uniform, and you can talk about differences induced, for example, between these subpopulations. And then we'll study all the additional information, say disease information, relative to these clusters of images. So we've written a mathematical model of this, fitted to the data. Let me show you one example. In a large collection of anatomical images of the brain, so I'm showing cross-sections, we detect 3 distinct templates that represent 3 subpopulations. And so if you look at those, you can see very clearly degeneration of the brain and enlargement of the ventricles, but also degeneration in this area. We call them young, middle, and old, because if you look at the histogram of the ages, so for example here for 3 populations, we can see that there is a histogram of ages that is very young, although there is a secret to staying young somehow that some of these brains discovered. And then 2 modes that are quite similar in their distribution of age, but then when you run regression on Alzheimer's, and well, so here it's very early Alzheimer's called mild cognitive impairment, then we discovered that actually if you are in cluster that is drawn here with red, you have twice as much chances to develop Alzheimer's as the one in yellow. Interestingly enough, this model also doesn't just explain the age uniformly. It chooses to spend quite a bit of complexity at the advanced age, which sort of, one of the speculations is that perhaps there is more variability at the advanced age, and you do need more complexity to actually explain it. So to conclude, this particular project sort of reformulates the problem and allows us to reason much more precisely about anatomical differences within the population and how disease is affected, which will hopefully lead to better understanding of the disease and better therapeutics. Thank you.